Good morning, everybody. May peace be with all of us today. I am Stais Norma Etika, currently an assistant professor at Magister Study Program of Clinical Laboratory Science, Universitas Muhammadiyah Semarang, Indonesia. Thank you for the time given to me to deliver this keynote speech for the 7th International Conference on Biomedical Engineering and Pharmaceutical Science, or ICBEPS 2022, held by the International Association of Applied Science and Technology, or IAAST. My presentation today is entitled Potential of Marine Bacterial Fibrinolytic Protease Enzymes as Antithrombotic Agents in Medical Devices. This topic is related to the research roadmap that I am currently going with a team consisting of clinical laboratory science and technology students, lecturers, and collaborating research from other institutions. The work that I am presenting today was started in 2019, research on antithrombotic agents, which will later be applied to medical devices, initially supported by a mono-year postgraduate thesis grant for my student. Yet, since the results were quite good and extended works continue to be done. This is a collaboration work conducted with the Biotechnology Center of the Ministry of Maritime Affairs and Fisheries, which now part of the National Research Agency, as well as Indonesian Science Institute, or LIPI, in Mataram. The collaboration was regarding to the sampling of sea cucumbers from which the bacteria were isolated. My goal in presenting this roadmap is to show the journey of research from scratch, from having nothing, making it a snowball, and supporting other research with my non-academic background. From the top to bottom, the roadmap looks like simple to be more sophisticated. Let's go to the idea of research on antithrombotic agents from marine bacteria on medical device, starting from how this research idea was obtained. Basically, the idea arises because of the global concerns or problems in the world in health. Sources of information to get this idea were from data of WHO, or World Health Organization, Ministry of Health, and the National Research Master Plan. There are also journals, magazines, scientific discussions, newspapers, and the experts. In particular, it's important to know the top 10 areas that our national research priorities focus on. Meanwhile, the roadmap for antithrombotic agents use marine materials because the marine environment actually is the priority focus area in Indonesia, in addition to health and medicine. So, in my opinion, the more priority areas of the government that are included in the research roadmap, the better, because they are important yet unique. More specifically, the idea of concern for epidemiological issues as well, because the disease causing the highest mortality in the middle and advanced economic countries throughout the world in 2016 was CVD. 30%, for example, coronary heart disease, atherosclerosis, ischemic, while the main CVD morbidity factor is the occurrence of thrombosis. Thrombosis is the occurrence or the formation of blood clots that clog blood flow and thereby blocking the flow of oxygen to the heart. On the other hand, the sea offers a very high novelty of material compared to the land, because 70% of the Earth's surface is ocean and the seabed is rarely explored. While Indonesia's tropical ocean biodiversity index is very high, number 2 in the world, it's very possible to get new materials that other countries do not have. Then there is the scheme here. Genetic engineering technology offers the possibility of mass production of proteins and secondary metabolites of living things, with living cells as the engine. In the future, bacterial enzymes with the best characteristic will be reproduced through recombinant technology or molecular cloning or genetic engineering based on the target gene encoding the antithrombotic enzyme from this wild-type bacteria, which can be inserted into vectors and then into competent bacteria so that genetically modified organisms can be obtained. Then the problem of occlusion here in the corner of this slide is the blockage of blood clots in medical devices that are implanted into the human body with various functions such as delivering oxygen, food, and medicine. This is an important issue that needs to be solved in the context of CVD, because the occlusion causes malfunction of this medical device. The combination of all the opportunities and challenges of these threats give the rise to the ideas contained in the title of my research that I presenting today. Let's go to the scheme here in the right part of this slide. The importance of the research that I am presenting. From the sample is from the sea, and the novelty of bacteria. If new bacteria have new enzymes, then there is an opportunity to use molecular methods to identify new bacteria. In my opinion, pathogenic agents from new groups of microorganisms are a real threat in Indonesia. The World of Health, for example, shows that SARS-CoV-2, when present in a host, becomes a microorganism. The virus is proven to easily mutate into new variants, and this is very dangerous because it becomes difficult for the body immune system to recognize. It's important for us to know how to identify molecularly new microorganisms in general, because besides being important, the workflow is also more or less similar between virus, bacteria, fungi, because it involves PCR sequencing followed by bioinformatics analysis. Here I use the term agent, for example, antithrombotic agent, to broaden its use. Agents will not always be in the form of drugs, but also include biomaterials in medical devices or reagents in diagnostic methods. I prefer to open all of these opportunities. Let's talk about the novelty. The reason why it's important to isolate materials yourself is due to novelty concerns. Don't let us be in the middle of the road, and be careful that the materials used probably already are claimed by other parties. This may hinder the continuation of the research or production in the future. So in my opinion, it's very risky if our research use commercial materials. So it's better if we use the native or from the origin that nobody has done before. Does that mean if it's made from natural materials, the isolation will take a long time? So this question is quite often addressed in terms of isolation. So the reality is, from my experience, not even so, because the support of today's knowledge, the process of isolating bacteria, enzymes, chemical compounds can be done easily, even in the lab. The stage may be many, but tend to be easy, and the many stages are opportunities for mastering tools, methods, or skills for our students. This has been proven in student research that on average students can finish on time with skills including bacterial isolation, microbiology, colony purification, gram staining, etc. Biochemistry competence of enzyme activity testing with spectrophotometer, hematology skills for anticoagulant, antiplatelet testing, and molecular biology skills at this stage can be mastered by students. So I move to the third slide, getting closer to the background or the determinants why this research should be conducted. The data from American Heart Society and Southeast Asia report in 2016 shows that in the non-infectious disease groups such as cancer and diabetes, CVD or cardiovascular disease still ranks as the number one cause of death in the world. The use of medical device is one of important efforts in overcoming deaths due to CVD. However, there's a problem of partial or complete occlusion, as we can see here, such as thrombus blockade, which can cause the device to malfunction. So we have entered into the more specific problem now. Yes, the problem of occlusion in medical device. This problem needs to be solved by conducting research. The upper right of this slide shows the mechanism of occlusion in medical device. It begins with the addition of plasma proteins such as fibrinogen, fibronectin, followed by the attachment and activation of platelets, which causes a kind of entrapment of red blood cells, leukocytes, so that all of them form a thrombus. The picture below illustrates 4 types of occlusion due to thrombosis of medical device. This slide contains the 4 objectives based on the background I presented in previous slide. These objectives was determined as well as results of analysis of the existing problems. In setting goals, I apply a strategy called SMART. So S means specific or right on target, M measurable, can be measured, A achievable, can be achieved, and R realistic, according to available resources exist and time bound, meets the deadline. The purpose of the research, according to the title, is to get an antithrombotic agent in the form of protease enzyme from marine bacteria isolated from sea cucumber in the sea waters of Nusa Tenggara, then later produce it on a large scale through recombinant technology with competent cells, and then the use of bioreactors for the term of raising recombinant bacteria, then recombinant enzymes productions, harvesting, purifying, and then immobilization on graphene oxide nanomaterials. Graphene oxide is a material commonly used for enzyme immobilization and is widely used in medical devices. My goal points are colored in yellow, and the blue ones are the implicit target of mastery of tools, skills, and technology. In this literature review section, I'm just emphasizing here several SWOT analyses. However, there are important mechanisms that need to be conveyed. For example, the possibility of enzyme immobilization on graphene oxide in the image above right, as well as the group of antithrombotic agents consisting of heparin compounds. The size is relatively small compared to the other group, namely enzyme. Here is heparin, here is the enzyme. We can see here the size is relatively small. The problem is heparin could not be immobilized but enzymes can. These concepts support the background as a situation analysis, which can also guide the method we choose. The next steps to take is to manufacture the enzyme and to do the test on the interface of nanomaterials, surface modification, and then evaluation of antithrombotic activities. At glance, from the roadmap view here, parts are in Bahasa Indonesia. It can be seen that the aim of this research is to obtain an antithrombotic agent in the form of enzyme that can be used on medical device through immobilization. What is the stage? Actually, currently in progress for the second year. So for application on medical device, maybe still in the next 4 or 5 years. In this slide, I have projected the number of working students, the method, as well as skills required to the project that is going on in my lab in the first to third year. The next step in the near future is to conduct whole genome sequencing, enzyme immobilization to all tests related with the safety of the use of thrombolytic protease for human. So the year 1, 2, 3 have already conducted. Some have been published, some are in the progress. The main result of the first year research in my lab include the finding of Bacillus stichilensis being able to produce crude fibrinolytic protease enzymes that are able to lyse blood here, lyse blood clots, and the activity was better than the standard nattokinase. Nattokinase is a commercial fibrinolytic protease enzyme produced by Bacillus natto, discovered from the fermented soy natto products in Japan. Another result in the first year was another bacteria called Bacillus aryabhatae, which has the ability to cause thrombolysis, and then has anticoagulant and antiplatelet activities at the same time. Therefore, they could be called antithrombotic bacteria. They have antithrombotic characteristics. The pictures below show the result of each stage of the research, and I think there is a sea cucumber here, and then the clear zone showing the enzyme production by the bacterial colonies, and then enzyme extraction resulted in and the results of anticoagulation, antiplatelet, and thrombolysis tests, as well as molecular identity and bacterial phylogenetic tree. And summary of publications obtained in the first year of my research. There are 5 reputable international publications indexed by Elsevier and Web of Science. Results of our research in second year are presented in this slide. Most of them are in the process of publication. Since the data has not been published, I could not share. However, I can say that the partially purified bacterial enzyme using ammonium precipitation and dialysis resulted in 3 times higher enzyme activity than that of crude extract. Our enzyme has optimum temperature at 50 degrees Celsius. It is expected that the thrombolysis, anticoagulant, and antiplatelet activity of the enzyme could be increased or higher than positive control after purification. Currently, the specificity of our bacterial protease is being characterized by zymographic method. And our next step is further purification by chromatography and ion exchange 1. So these are the references, including our published papers related to the project that I presented. Finally, I would like to thank you for all of your attention, and I would like to show my deepest gratitude to the committee for the opportunity to give this keynote speech. I hope you are happy and healthy. Thank you.